we have with us Dr. Gagandeep Kang. Many of you, almost all of you perhaps know all about Dr. Kang, and the address for the topic now is At War with Viruses, How Science Can Beat Viruses. And she's a professor in the Department of Gastrointestinal Sciences at the Christian Medical College, CMC as it's called, in Vellore, Tamil Nadu, India. She's the Vice Principal for Research and Head of the Wellcome Trust Research Laboratory at CMC. And Dr. Kang's research on pediatric diarrhea focuses on rotavirus epidemiology, prevention, and vaccine development. And for molecular strain surveillance, she's built her own work to develop a network of India-centric virology laboratories and the WHO Rotavirus Reference Laboratory for SEARO, all of which have resulted in insights into the epidemiology, burden, and transmission of rotaviruses in India. Dr. Kang, it's over to you.
Thank you very much. I'm going to be talking to you about how we use science to talk about viruses and what it is that we can do. I hope that you can see my slides. And I'm going to use as an example the virus that we've lived with for the last couple of years and not really draw upon the work that I've done before this. As with practically every other virologist in the world, we've also been drawn into studying COVID-19, but today I'm not going to be talking about our own work. So if we look at science and viruses, everybody talks about how science brings us solutions, technology brings us solutions, but the solutions and the learning just keep coming. We've had some really exciting news in the past week because Moderna announced results from their bivalent booster candidates. So they looked at a vaccine, their original mRNA vaccine, which was made based on the older strain of the virus. And then they made a vaccine that was based on the beta strain of the virus. And in their announcements, as you can see here, they claim to have met the objectives that they had for their bivalent booster candidate. Now, just to show you the science behind this, this is what it is. What were they trying to do? They had an old version of the vaccine, which is called mRNA 1273. They also made a new version of the vaccine based on the beta strain, and they mixed the old and the new variants together, the mRNA for the variants together, to create a vaccine that they called mRNA 1273.211. So if you look at the top graph, I'm going to walk you through the first figure so that you get a sense of what we are talking about. So in the first column, what you see here is mRNA 1273, the original mRNA vaccine. And in the next, you have the mRNA 1273.211, 50 micrograms. What do you see here? These are people who had received 2 doses of the Moderna vaccine at least 8 to 9 months before. Their antibodies, their neutralizing antibody titers before they got a booster dose were just above 100 on average. And then after they were given another dose, a half dose, the old dose was 100 micrograms, they were given a half dose as a booster. This then went up at 4 weeks after immunization to over 1,000, 1952. And then at 6 months, it declined to 673. When you compare the old version and use the same 3 time points with the bivalent booster, what you see is you started at a similar sort of level, 150, 103. You boosted to about the same level, but you were able to maintain antibodies at a somewhat higher level with the bivalent boost than with the homologous boost or the same vaccine being given again. Now, this was for the ancestral SARS-CoV-2. Now, if we come down and look at beta over here, this is beta, and this vaccine consists of a mix of beta and the old version of the virus. What you see here again is that when you have the combination gives you better results than the ancestral version of the virus. Not only do you see results against the two viruses that are included in the vaccine, you also see a pretty good immune response in Omicron, which is not related to the ancestral virus or the beta, and for Delta. These are lower than we saw for the ancestral strain or for the beta strain, but still pretty good. On average, anything above 100 that is sustained over time is considered pretty good. So if you look at these figures, what are they showing us? They are showing us that when you give a bivalent booster dose of the mRNA vaccine, you make antibodies that are better than antibodies if you use just one virus alone, and that these antibodies last for a reasonable length of time. In addition, you also see that the breadth of the immune response is higher because wherever you give the bivalent performs better than the ancestral way. So this is the science that we have this week. What does this mean for us? This means when we begin to think about booster doses, maybe we are talking about the next generation of vaccines. Is this the end of what we will learn? I don't think so. I think we are going to keep learning as we live with this virus and we continue to investigate what we can do with modifications of the virus and also what the virus is doing in humans and in populations. So let's go back now to January of 2020, where we had the first description of SARS-CoV-2, which, it's a virus, everybody knows that, and the virus has been described as a piece of bad news wrapped up in a protein many, many years ago. And if we are to study the virus, it's very important for us to have the genome of the virus, because when you have the genome, you can understand what structure is, what function is, and how to modify it. And this description that has been published in the New York Times is a great way of trying to understand what the virus does. This is a very large virus, 30,000 RNA letters in its genome. There are 16 proteins in the virus that don't actually make up the structure of the virus. There are only a few proteins, the S, E, M, and N proteins, which are structural. Everything else is actually functions that allow for the virus to replicate within cells. And for some of these proteins, we understand what the function is. For many others, we don't. NSP1, for example, suppresses an antiviral response. NSP4 creates the vacuoles within which the virus replicates. NSP12 is involved in the copying of the RNA of the virus in order to make proteins. NSP5 clips different parts of the genome that is being read so that you have sequences for proteins to be made. There are also many proteins for which we don't quite understand what the function is at the moment. So when we were at January 2020, what did we have? We had a number of scientific challenges. We had a lot of logistic challenges, but the first one was to know, did we have the right tools to be able to identify the virus? And if you think back to April of 2020, one of the biggest questions was ICMR made a recommendation of using antibody tests in order to be able to detect clusters of infection. And this was something that horrified the medical microbiology community because the tests were not very good, and using antibodies to diagnose clusters is not really the best epidemiological tool to have in an evolving situation. We are in a much better position now. We were able to use the sequence of the virus to create very rapidly RT-PCR tests. We were able then to develop antigen and good antibody tests. And we're now in a pretty comfortable position where diagnostics is concerned. We also had to begin to think about treatments. And for treatments, the first thing that you do is try and see if you have drugs that are already available that you could use. So chloroquine was an example of an available drug that at least theoretically could be used. You also think about new drugs. You can also think, in the case of viruses, we know that antibodies really matter. So could you use antibodies from people who had been infected, or could you develop new antibodies that could be used for people? And one of the remarkable things about this pandemic has been not just the amount of work and studies that were done, but the coming together of people to be able to use the right kinds of tools in order to evaluate treatments and vaccines. Particularly the studies, the trials that are called Recovery and Solidarity are the scale of trial that has never been done before. It's actually advanced clinical trial science, because we've had adaptive trial designs, where if one treatment is not working, you can take it out and replace it with another treatment. So we've done a lot of work, both in terms of preclinical as well as in clinical evaluations, to be able to come up with drugs. We also had challenges in vaccines where we had untested platforms. We were trying to work at a speed and a scale that had never been done before, with partnerships that between regulators, the government, companies, academics, that had also never been done at this scale before. And of course, regulators had to interact with each other and with WHO to try and figure out how to get vaccines to us that were safe, effective, but also rapidly available. Now, it's important to remember that all of the work that we've done in the last 2 years actually builds on work that had happened for many, many years before that. We've actually known human coronaviruses, and the ones that were in humans before 2000 were the coronaviruses that generally cause mild upper respiratory infection. But in 2003 and subsequently, we've had coronaviruses that caused serious disease. SARS-CoV-1, as it's now known, as well as MERS coronavirus, taught us a lot about infections that we were able to use during this pandemic. What we did know from SARS-CoV-1, and I'm not going to walk you through this entire figure, but just to say that we understood that viruses needed to have receptors that allowed them to get into cells, that once viruses got into cells, their message had to be read to make a number of proteins at different locations within the cell, and then the fully formed new viruses have to be released. Now, this is a process that is followed for all viruses. And the advantage of having an understanding of the replication of a virus is this allows you to identify spots for intervention. So the more you know, the more you can think about where are the points where I can interrupt the replication of the virus. Now, of course, the first thing would be, can we stop the virus from getting in? And from the previous coronavirus studies that had been done, it was very quickly possible for us to think about how SARS-CoV-2 replicated as well. The ACE2 receptor had been identified for SARS-CoV-1, but there was a second receptor, which is called TMPRSS2, which was being used by SARS-CoV-2. And the way this worked was that you have a spike protein that binds to the receptor. You cut off the top of the spike and bring the virus and the cell close together. They fuse with each other, and then the virus sends its message into the cell. This whole process takes about 10 minutes. So if you have to interrupt any of this happening, you have to be able to be present before the virus even gets in. And sometimes this is what you can do with preformed antibodies. But there is more than one entrance mechanism. So if you want to block viruses, you actually have to block all the ways that the virus can get in. And I had mentioned chloroquine previously, so I think it's important to point out that chloroquine actually blocks endosomal fusion, which is one of the ways in which viruses can get into cells. Now, Omicron actually uses endosomal entry and is not temperature dependent. So the question arises, if we used Omicron, if we were studying Omicron infections, if that had happened before all of the other variants we saw, would chloroquine as a treatment have worked? Well, those trials are no longer being done now, but we do know that chloroquine didn't work for the older versions of the virus that we had. So if we think about therapeutics, we can also think about, where else can interruption happen? And one of the things that is being very widely used is a variety of monoclonal antibodies that try to prevent the virus from getting into a cell. And as we've seen with Omicron, most of the monoclonals that we had stopped working. So what had worked well against Delta, shown here in blue, did not work against Omicron, which is shown here in red. There was one monoclonal antibody, sovitramab, that worked, but that worked only for BA1 and didn't work for BA2. So understanding what you're dealing with and what treatments need to be applied at what times, in which patients, requires a deep understanding of what is happening. So therapeutics, understanding, as I mentioned, structure, function, replication, allows us to then develop a variety of new drugs. And molnupiravir and Paxlovid have all been developed based on a deep understanding of how the viruses replicate. Steroids, similarly, we now know that steroids work by reducing a hyperimmune response. This is also the reason why you have IL-6 that is involved in the generation of an inflammatory response. Many of the drugs that target IL-6 can also be used in SARS-CoV-2 for this reason. Now, when we come to vaccines, a deep understanding of viruses becomes even more important. Earlier work on MERS and SARS had shown that in the spike protein, where we understand the structure, we needed to have a stabilization of the spike protein in order to induce the right kinds of neutralizing antibodies. And this data built on work that had been done looking at respiratory syncytial viruses, not just, so we had work on HIV, RSV built on the HIV research, and then SARS-CoV built on the RSV research, and SARS-CoV-2 and MERS built on the SARS-CoV-1 research. So this proline stabilization has been used in multiple vaccines in Moderna, Pfizer, Novavax, and Johnson. And now we are developing a 6-proline stabilization that might be even better than the vaccines that we have at the moment. We also need to understand how the antibodies work. There are 4 classes of antibodies that are responsible for neutralization of the binding between the virus and the ACE2 receptor. And then depending on which one you have, they can be highly potent, but they will also be in the regions that are most targeted by mutations when we have variants of concern arising. So there are advantages and disadvantages to making different kinds of monoclonal antibodies. All of this understanding of structure and replication allows us to understand what the best points of intervention really are. And understanding sequence data also tells us a lot about the variants that can arise, why they arise, and why it might be very difficult for us to get rid of this virus. We know that this virus can spread across species, and that makes elimination or eradication of this virus very challenging, because you've got a number of host species in which this virus can go and hide and then evolve through combination with human coronaviruses. Now, we have data that has been generated from sequences, from the lab, etc. But I think ultimately data has to come from humans. And it is the ability to do studies that are in cohorts. What this shows you is essentially what happens in terms of neutralization in people that are infected with different kinds of viruses. And essentially all this figure is showing you is that Omicron, which is shown here in black, it doesn't matter what kind of infection you've had previously, Omicron could not necessarily be blocked easily by pre-existing antibodies. A fact that was available in lab studies early on using samples from humans that was then validated in the epidemiological information that we had. So to end, we had some exciting news in the past week, but with science, there's always exciting news because there are always questions that remain to be answered. Just for SARS-CoV-2, what about cellular immunity? How long should you take for boosters? What about long COVID? When you have drugs, when will antiviral resistance come? What really are the best combinations that we should be thinking about? There is no end to the questions, but I think together, what we have shown in the past 2 years is that we can answer a lot of questions really fast, and I hope that we will continue to do so. Thank you.
Thank you so much, Dr. Kang. We have a couple of questions. I wonder if you would be willing to take them. Just for those who, before that, just for those who have just joined in, there was an extremely enlightening keynote from Dr. Kang. She explained to us how the work that Dr. Kang and others around the world have done in the last 2 years impacts what is going to come after, and what we've learned from the vaccine, what we've learned from how the vaccine has worked among people, what we are continuing to learn. That was extremely enlightening. And Dr. Kang, I just want to just take a question. Someone who refers to themselves as anonymous has asked a question which I think you mostly answered, which is, since when SARS-CoV-2 has been with us? Because I hear it was there before as well. You did show us that slide showing that it's been there for more than 50 years. But I will take the opportunity to kind of reframe that question a little bit. Does the scientific community at this point in time worry at all about what the origin is of this virus? Because that is also the question that this person has asked. Or is it something that you're saying, okay, this is a fact of life, and let's focus on what we can control? What's the approach like?
Okay, so let's look at it this way. Is SARS-CoV-2 a new virus? Yes, it is. Does it have relatives? Yes, it does. Which is its closest relative? It's SARS-CoV-1, which we had in 2002, 2003. Could this virus have been made in a lab? Sure, it's feasible. Could this virus have evolved naturally? Sure, that's feasible too. The closest relative we have for this virus is actually a virus from bats in the mid-1980s, where it was hiding away during this time. Whether it was being experimented on or was evolving naturally is not something that we can answer with the data that we have.
Right. Just a couple of more questions are there. And this is a Reimagining Healthcare Summit. But obviously, the audience, we're all experts here, but we're also people who have suffered through the pandemic. So there are some sort of basic questions which I'm going to take the opportunity to ask you. Someone asks, are all viruses bad for us humans?
Some viruses are actually an advantage for humans. So in fact, when you have an antibiotic-resistant infections, one of the new treatments new and very old treatments that people are trying out is viruses that infect the antibiotic-resistant bacteria. Those viruses are called bacteriophages because they eat up bacteria. So viruses can be beneficial as well as being harmful.
Correct. And that in fact was a question by Dr. Fuad Halim. And there's one last question, Dr. Kang. Someone is asking you to explain, and I don't want to kind of interpret that question. So I'm just going to read it as it is.
I've seen the question in the chat box, so I'll answer that. Okay, so with any vaccine and any drug, I think it's important to understand that nothing is perfect in this world, right? So with every vaccine, with every drug, you don't expect 100% efficacy and you don't expect 100% safety. With many existing vaccines, we know of side effects. There are common side effects, which is the pain, the swelling, the fever that you get when you get a shot. There are also rare side effects, and the rare side effects can vary quite a lot. Guillain-Barré syndrome has been associated, it's a paralysis, has been associated with some influenza vaccines. And with the AstraZeneca vaccine and the Johnson & Johnson vaccine, we have had a special condition that is called TTS, thrombosis with thrombocytopenia syndrome, which is the formation of blood clots and a reduction in the platelet count. This is a very rare side effect. In places where it has been described as a rate, the rate has been a maximum of 1 in 40,000 vaccinated people and as low as 1 in 2 million people. This is associated with the viral vector vaccines. The way you can look for it is to look for the occurrence of a blood clot as well as a reduction in the platelet count and the presence of an antibody called anti-platelet factor 4. If these occur between 4 and 20 days after a dose of the viral vector vaccine, it is likely to be associated with the vaccination. This is a rare side effect. It is known. Our AEFI system in India is a 3-tiered AEFI system, but has picked up very, very few cases of VITT or TTS. Most of the data that we have on this condition comes from high-income countries.
Okay, that was enlightening. And just a reminder for those of you who want to listen back, the recording is on. It will be available on YouTube as well. On that note, thank you so much, Dr. Kang, and thanks for taking those questions as well. There's plenty to come in the summit, exciting conversations, but this was a great start. Thanks.